Here we go. So these are pictures from the sampling event we did this past spring in 2022. Swan there, a tundra swan over in Harrison Slough, which is just to the east of Coeur d'Alene, Idaho. Another one there on the bottom that had just been collared with a black collar. And then myself and another person sampling feces in Hepton Lake, which is a reference area that we went to, and then our laboratory at night. And before I move on to exactly what we did, just wanted to mention all these folks that have been involved with the project. And I've got data basically presented from each one of these groups in this presentation. And so, yeah, I'm just convinced that we can learn so much from poop. I did a whole feces thesis with my PhD and sage grouse, looking at poop and stress hormones and things like that. So I've carried it on into my regular career, and I think we can learn lots from it. And it's easy to collect, and EPA will let us do it. Capturing swans is a little bit more tricky with EPA. And so the first thing we did is to do DNA sequencing from the fecal samples that we collected, all towards the effort of understanding how lead exposure varies across the site, across the lower basin to the east of Coeur d'Alene, Idaho, trying to figure out if plant species affect lead concentration, lead exposures. So using the RBCL marker in fecal samples, trying to make sure that we actually collected the right sample. Was it a Canada goose or was it a tundra swan or something else? So the cyclooxygenase marker there, and then the CHD for sex determination. We also used the fecal sample collected there in Idaho to look at lead isotopes. And so that's been used in lots of different situations. And it's really nice in Idaho where the lead is stopped fractionating from 1 billion years ago. So the lead signature in the Bunker Hill area is quite unique. So that really helps us figure out that we've got a Bunker Hill exposure versus something else. And then XAS is X-ray absorption spectroscopy, which tells us what kind of lead species it is. Some lead species are really bioavailable, others are not. And so this here is a galena, which is a lead sulfide. It doesn't really get into the blood of birds. It's more the organic forms that get into birds. So that's the approach. I'll get into the details of the study in a second, but background here in case you weren't aware, back in the 1800s, mining began in this area. And until 1968, all these tailings were dumped into creeks and stream beds and things like that in the upper basin that had then moved down into the lower basin where we're working. 100 million tons of mine waste, 2.4 billion pounds of lead dispersed over tens of thousands of acres. A real mess. The blood lead levels in people in the residential areas after being exposed to kind of fallout from that bag house explosion were the highest in the world. And so this is the lead kind of contamination coming down the river systems, down the Coeur d'Alene River into the lateral lakes that then sediments into the areas where the swans come every spring. And this is from Craig Ely's work with PTT tags, and doesn't represent everything that he's done, but it just basically shows that there is a location that the swans come every spring, that they get exposed to this contamination from the mining activities. And up close and personal, this is the lower basin. The Coeur d'Alene River is working its way down through all those red highlighted lakes. Those are the lateral lakes where we did our study. And where we are in the Superfund process, I've mentioned this project to a few people that have been working on lead for years. Chris Franzen, somebody I worked with quite a bit at the USGS, asked me if this is still he couldn't believe this is still a problem. Everybody knows Chris and keeps track of things really well. It's surprising that it actually still is a problem. 1983 is when we started, but we've mainly focused on the human health impacts. And so that's where we've spent most of our time up until sort of current, where we're now working on the remedial design and especially remedial action down the lower basin where the wildlife are exposed. So EPA managers and other sort of environmental quality folks would just wonder, why don't we just measure the lead in the sediment? Why do we have to look at the fecal samples? What's the difference? Well, first off, I don't have to tell you guys, but feces represents the specific exposure to lead in sediment. And the picture on the right-hand side, there's a couple of colleagues sampling lead and other metals in sediments, and it takes a lot of work as well. So it's not as fast and easy. Sediment doesn't necessarily represent the specific exposure, given these variations across the wetlands and bioavailability. You know, how much lead sulfide do we have versus lead citrate, for example. And we don't know exactly where the swans have been feeding perfectly, so we don't necessarily sample in the right spots. The swans do the sampling for us, and it's much faster and easier. And then you can find out all those other things that I mentioned in the previous slide. And I don't know if it's necessarily the only mechanism of lead exposure, but it's definitely considered to be the primary mechanism of lead exposure, which is the incidental contamination, the incidental uptake of sediment as the swans are foraging for, you know, water potato and other vegetation in the spring. And that work has primarily been by Nelson Beyer here, a paper from his looking at sediment ingestion, basically acid insoluble ash, a measurement of sediment, and correlated with lead in the feces. And so it's a pretty decent correlation between the two. However, you know, swans consume, and I guess to back that up, swans consume, we think, a fair amount of water potato. And to get that, you need to burrow into the sediments and expose yourself to sediments with lead in them. The water potato doesn't have a lot of lead in it. What else? There's large variation in sediment contamination and sediment uptake across the different wetlands. So is it this variation in sediment uptake into the bird that is responsible for this variation in lead exposures across the landscape? Is it bioavailability? Fair number of questions. It depends on the types of vegetation that they're consuming. So water potatoes might expose them to more incidental ingestion than, say, Eladia. But we're working on this sort of ongoing theory of exposure and addressing questions based on that. So first off, you know, developing this metric, envision this site, which is tens of thousands of acres. We don't have all the money in the world to fix every single one of those red wetlands. So we have to pick which ones we want to fix. We also have to figure out if we can't fix them, instead maybe create wetlands in different areas to pull birds to clean habitat. And we can't necessarily go and sample the sediments from all of the different wetlands and relate that back to the swan health. And so we want to know whether or not fecal lead mimics sediment lead concentration, and then secondly, origin. Because if fecal lead just mimics sediment lead concentration, then we can just sample sediments. But if it doesn't, we need to go out and design studies to use fecal lead monitoring program. Secondly, does fecal lead represent blood lead when looking only at Bunker Hill lead? So how meaningful is the fecal lead sample when it comes to what actually is biologically hazardous, is the indication that you get from blood lead? So is there a correlation there when only looking at the Bunker Hill lead using isotopes? Third, does diet influence lead exposure? I've already talked about this a little bit using the eDNA approach. And then how do swans use the site? Because we don't necessarily know how much they're moving between these different wetlands, and therefore how that will affect our remedial action restoration approach. So we did some of that. We looked in the Coeur d'Alene River watershed, which is where the lower basin is, compared to the St. Joe River watershed, where there's much less lead contamination. Intended to, and this is where I've been interested in what other people have been talking about today, is we use the airboat night lighting design. And I think what we basically did is we scared them away and captured only 17 of our 40. So we had 40 birds. We were looking for these different measurements here. We added some five GSMs and then 35 colored colors. Instead, we got 3 sites, just the control, low and high. Thankfully, we did get variation in the sites, but only 17 birds, and then 5 fecal samples with blood, and then 2 fecal samples later from birds that had died that we were later captured or picked up, and then 2 bone samples, 2 liver samples, and then 17 body condition indices. Now, I don't know if others have seen this, but we didn't see any association with any hematology in our physiological measurements. The lead did not seem to predict hematocrit for some reason, which is surprising to me. And then 29 wetland fecal samples, 9 sediments, and then trackers on 2 out of 5, and then 12 bands. We first wanted to mention that we actually did verify that we were successful in identifying tundra swan poop. They tend to commingle a fair amount with Canada goose, and we were able to pick it out using that approach there. So that was really good. I want to see if the sex of the animal has anything to do with lead exposure. or the diet they ate. We haven't looked at that data yet. So entering the first question, does fecal lead mimic sediment lead? And so this is, to me, it says the answer is not necessarily. The wetlands on the bottom there, Hepton and Robinson, could considered be sort of a reference location, lower lead concentrations. Those gray bars are the sediment concentrations. Schlepp, Strobel, and Thompson are the three areas that are contaminated, and those have varying levels of higher sediment contamination. But if you look at the brown bars, they don't always line up with the gray bars. So Thompson, more contaminated, but in terms of sediment, the fecal samples indicated a similar exposure to lead compared to Schlepp, which is less contaminated. And then the blood, it just indicates to me that the birds are flying around quite a bit and getting exposed in both the reference area, Hepton, and then also in the lower basin. And in terms of origin of lead, primarily Bunker Hill. This is, I'm not going to show the specific isotope data here, but primarily Bunker Hill lead. As you can see, they're 90+% in all three media. The Hepton birds, which are the reference site, flew back and forth. I think that's explains why they've got 70% Bunker Hill lead in them. So conclusion from that is that blood sampling indicates a broad exposure pattern to Bunker Hill lead. They didn't have a lot of lead from other locations. So that was kind of a nice thing to see. Fecal sampling indicates wetland specific exposure, and sediments are a general measure of possible exposure that could be used for things other than biomonitoring. So this one, is there some biological relevance to the fecal lead measurements? Based on just 5 birds, unfortunately, the answer would be potentially. And I think it's because if you look at these 2 lines here, the parallelism to them is striking, and that's primarily because it's all Bunker Hill lead. They're not really, the lead that they may have received in the southern parts of their flyway isn't showing up. And so had these curves been there been a mismatch between the two, perhaps, the blood lead that they had taken from somewhere else would misalign the 2 curves. Because the blood lead is primarily Bunker Hill, they're parallel. And I think that if we want to focus on, maybe coming up with a threshold for fecal lead and using, say, 50, we'll have to kind of figure out what our blood threshold would be. It might be above the 0.2, which is the Payne and Franson value. Our fecal leads might be somewhere, definitely below 20. So this kind of provides a little bit of biological relevance, but we need more data. Oh, excuse me. Does diet influence lead exposure? So remember, it was we think it's via incidental sediment ingestion. And we have to always, looking at the different wetlands, they each contain different kinds of plants. However, the sediments are primarily, 5,000 to 10,000 ppm parts per million lead. So there's not as much variation in the lead contamination as there's in the foraging strategies across the wetlands. And so that's going to differentially expose them to the sediment. And I say these are tentative data. This is using the RBCL marker in the Hepton birds on the left, the Schlepp in the middle, and the Thompson on the right. I think they're tentative because the connection between biomass consumed and DNA read count in the fecal sample we don't have that quite validated yet. So these are the patterns that we're seeing, and they're preserved across each bird. Each bar is a bird. And so it's relatively consistent. So we'll see once we have a stronger assay from year's work next year, we'll see how this lines up. But this is interesting. Hepton is dominated by burr reed. Schlepp was dominated, which is a kind of a medium restored location, Eladia. And then Himalayan horsetail was the primary food consumed by the swans at this really pretty nasty location called Thompson Marsh. And I don't go into what that might mean in the next slide here. Just recall that Thompson and Schlepp, the 2 groups, had the same fecal concentrations, but the Thompson had a much higher sediment concentration than Schlepp. So given that, the consumption of horsetail at Thompson might be less risky, maybe not as nutritious as water potato than consumption of Eladia, which was big at Schlepp, and other plants consumed at Schlepp. Perhaps because just the way that they're consuming these plant matter, they're not exposed to as much sediment. Secondly, maybe there's differences in bioavailability at Schlepp compared to Thompson. Schlepp has newer deposits of lead. It's not as anoxic as Thompson is, and so that can influence the type of lead that's there. That might make it more bioavailable. And then lead may be less bioavailable in conditions that favor horsetail growth. So we're going to look into these issues here. And then further work, as I mentioned, we want to validate our assay a little bit better than we have now to make sure that we are using relative reed abundance in the most proper way. And then assess bioavailability in sediments in conjunction with selected plant communities and lead concentrations in different plant types. And so there is some preliminary data, Schofield et al. Presented, that the tubers have really low concentrations of lead and leafy plants, not so much. I will move on. This is basically telling us that the green bars shrink in the fecal samples, which means the swans are eating the organic lead and they're not eating the inorganic lead. The lead that's bound to iron and other things like that. That's all that is saying. And then this would be interesting thing to finish on here. Where did the swans go? This is the swan captured at Hepton, moving all over the place within the St. Joe Basin. And then it is currently just sort of next to Edmonton. It flew up to Western Alaska. And as of, I guess, yeah, the 20 yesterday, it's right here now. The interesting thing is that, that lead concentration was 0.17. So we weren't super surprised that it did well. The second one captured at Schlepp did this, then went on its way to a little bit further south than the Hepton bird. This had a 3.8 part per million lead concentration, which is considered pretty high. And that bird actually has survived so far to be currently where it's at as of the 23rd. And these are just the next steps you guys can read through. I'm gonna let you go. Consider more effective. So basically in summary, blood lead indicates a 30-day turnover rate. So we're kind of measuring exposures across the lower basin. Feces represents a 6 to 12 hour turnover rate. So we're measuring wetland specific. And then sediment is obviously stable and more of a biogeochemistry question. And the rest of those I've already mentioned. And we're hoping to use this approach for sort of long-term biomonitoring at Bunker Hill as we restore the area. And I also want to get some tips on airboats versus other ways to capture birds in the spring when they can fly away from us.